This video will be dedicated to the idea of active transport, and we start with the concept of ATP. Now, this is actually a structure that was discussed in Unit 2, but I wanted to place it here because I thought it just fit into here a little bit better. ATP, we already had enough information in Chapter 2. So we're sticking this in here because I think it goes pretty well. So this structure that we're looking at here is called ATP. ATP, adenosine triphosphate, is the energy currency of the cell in almost all cases. There's a few cases that are a little bit different that we'll discuss down the line, but most of the time when your body needs energy to do some kind of a cellular some kind of a job within the cell, oftentimes it will use ATP. So we have adenine, ribose. This entire structure here would be called adenosine. Look at this. 3 phosphate groups. Didn't I tell you they were important? So the first one we're going to be looking at here is adenosine monophosphate. It would be adenosine plus 1 phosphate, monophosphate. Here we have adenosine diphosphate, adenosine plus 2 phosphates. And notice that ADP, adenosine diphosphate, has energy. And the reason it has energy is that the phosphate groups carry a negative charge in this case. And that negative charge, these guys don't really want to be right next to each other. Thank you very much. Negative things don't like to be right next to other negative things. Negative things are attracted to positive things. And so this phosphate group struggles a bit holding on to its bond with this phosphate. Yes, it's a covalent bond, but this bond is going to be weak. This bond right here is going to be weak. So we actually have a little wavy line. Wouldn't hurt you to, I don't know if you have to draw this entire thing, you guys, but you could just say like adenosine in one area, and then go ahead and draw those phosphates, draw the negative charge, draw these with integrity, draw these with faithfully. You want to draw them just as they are. It's not going to take that much work because it's super important to understand the concept of this bond. So this phosphate attached to this phosphate, it's a little bit shaky. It's kind of an unstable bond. And if this bond were to be released, it would release a burst of energy. Here we have adenosine triphosphate, and this is the full thing, the full shebang. And so you see what we've done here, of course, 3 triphosphate. 3 phosphates stuck together means that this is highly unstable. This guy really doesn't want to be part of this family. And this bond here is going to be super, super unstable. And once it releases, it will release a great burst of energy. And we show that here. I would also suggest writing down this particular formula. What we're showing, of course, is a hydrolysis reaction here. With this arrow, we are showing a dehydration synthesis with this arrow. So, we've seen this before. That's not the most important thing here. The important thing is that ATP can be broken apart into ADP, a molecule of phosphate, which is sometimes abbreviated PI, P sub I, which means inorganic phosphate. You can just draw a P with an arrow around it. That's fine. And energy. So as the ATP is released into ADP, an inorganic phosphate, as that hydrolysis occurs, it's going to release energy. So if you have, this is a lot to look at here, especially if you've never seen a sodium-potassium pump before, but this is the typical way that we show this. There's a lot to look at in this picture. I guess I'll just take you through it. Here we have an ATP that is attached. As you can see, this is the inside of the cell because it's below the plasma membrane. As the ATP is attached to this guy, it is in its low energy configuration, which means that it's basically this protein, it's called the sodium-potassium pump, there it is sort of facing, if you will, the inside of the cell. Now the ATP is going to be hydrolyzed. You're going to leave that inorganic phosphate attached, and it's going to be broken apart into inorganic phosphate and ADP, and as a result, it releases energy. That energy will actually change the configuration of this protein, and as the phosphate continues to be attached, it will face, as you can see, the outside of the cell, thus releasing these 3 little beads here, which happen to have been sodium ions. As the sodium is released outward, there's a binding site for sodium, but on the other side there's a binding site for potassium. So the potassium ions K pluses, are going to come along and they're going to bind to their little binding site as the protein is facing outward. And then look what happens next. The phosphate releases from its binding site inside of the cell, and as a result, the protein, the sodium-potassium pump, again faces inward. And then the next thing that's going to happen is that the ATP is going to be bound again, and sodium is going to come along, and then you're going to have this guy here. So in order to change the configuration from internal facing to external facing, thus allowing for this unusual exchange of ions, sodium and potassium, you need a molecule of ATP to reconfigure that protein. And I think the reason why this one is brought up all the time is because it's a super, super important protein in your body. If there was a toxin that came along and blocked the ability of the sodium-potassium pump to do its job, you'd probably be paralyzed within 10 seconds and dead within 30 seconds. If all of the sodium-potassium pumps in your body no longer function anymore, it would be immediately fatal. That's how important these guys are. This exchange of sodium outward and potassium inward is actually super important. So why is ATP required to do this? Well, it turns out that what we're doing is we're pushing these ions against their concentration gradient. Remember that when we discussed diffusion, we were taking substances and moving them down their concentration gradient or with their concentration gradient. Here, we're going against. Sodium ions are more highly concentrated outside of the cell. So they don't naturally move outside of the cell. They naturally move into the cell when they have an opportunity. And here we're taking this protein, this pump, and we're pumping them out into an area, from an area where they're less concentrated into an area where they're more concentrated. And the same with potassium. It's just the same. There's less potassium outside of the cell in the first place, but what we're doing is we're grabbing a bit of that potassium that isn't even that highly concentrated out here, and we're pushing it, pumping it into the interior of the cell, the intracellular fluid, where it is in fact highly concentrated. And in order to facilitate doing that, you need something more than simply a channel, or a tunnel, right? You actually need a pump to facilitate that, and that is why ATP is required for that. So ATP is hydrolyzed, releasing energy, which allows for that protein reconfiguration, the shape change. All right, there's two more physiological functions that we're going to be discussing. They both pertain to vesicular transport. So vesicular transport is where we use vesicles. Here's the spelling, vesicles. We use vesicles to move substances into a cell or move them out of a cell. So these vesicles will transport large molecules, macromolecules, and fluids across the membrane or within the cell. A typical way that we picture this happening is with cells of our body that are especially set up to consume pathogens such as viruses and bacteria. A lot of those would be like macrophages typically. And what they're able to do is simply consume things. Now this guy here, this vesicle, is going to be of the same substance. You can actually, if you look closely, you can actually see it's the same bilipid membrane that made up the plasma membrane. So we don't really draw it very well in these pictures usually, but you should remember that that is a bilipid membrane. So we have the bilipid membrane, it simply consumes things. This is actually what I was just discussing, phagocytosis, where you have a pathogen, you're taking it into the body. We'll talk about that more down the line. All right, and then here's a picture of exocytosis. This type of vesicle transport, substances from inside of the cell are removed from the cell and enter the external environment. They're actually much better portrayed here. You can actually see the bilipid membrane here. There's going to be a couple of proteins involved too. Don't worry about those too much. There's actually quite a lot of research still being done on the way that these things actually work. But so these membranes here will fuse, because they're made of the same material, they will fuse to the plasma membrane, and then the vesicle will push its substances out of the cell. There's an actual picture of that happening here. It's pretty cool.